The suprachiasmatic nucleus, SCN, is located within the hypothalamus of the brain. The hypothalamus regulates numerous bodily functions. The SCN acts as a central pacemaker, coordinating various biological rhythms. The suprachiasmatic nucleus, SCN, generates rhythmic signals transmitted throughout the body. The SCN releases signaling molecules that travel via hormonal and neuronal pathways. This mechanism synchronizes internal processes with the external environment. The suprachiasmatic nucleus, SCN, influences sleep-wake cycles and hormone secretion. The SCN also regulates body temperature and metabolic activity. Disruptions to the SCN can lead to sleep disorders, metabolic dysfunction, and other health problems.
Peristalsis involves rhythmic contractions and relaxations of smooth muscles within the intestinal wall. These wave-like movements propel food and waste products along the digestive tract. Peristalsis ensures efficient nutrient absorption and waste elimination. Smooth muscle cells within the small intestine contain a biological clock. This internal clock influences the timing of peristaltic waves in the small intestine. The biological clock helps regulate digestive processes on a roughly 24-hour cycle. Meal timing significantly impacts the intestinal clock. Regular meal schedules can synchronize and strengthen the gut's biological rhythms. Irregular eating patterns may disrupt the gut's biological rhythms, potentially affecting digestive health and efficiency.
The abdominal cavity houses several vital organs, including the liver. The liver is situated in the upper right quadrant of the abdominal cavity. Understanding the liver's location is crucial for comprehending its function and cellular activity. Hepatocytes are the primary functional cells of the liver tissue. Healthy hepatocytes actively perform metabolic functions, including glucose regulation. Close-up visualization of hepatocytes demonstrates the cellular mechanisms involved in glucose metabolism. The hepatocyte nucleus contains a circadian clock that regulates the timing of glucose release. The liver's metabolic activity is synchronized with the body's internal clock. This synchronization ensures a regulated supply of glucose to the body.
Adipocytes, or fat cells, are the primary cellular components of adipose tissue within fat depots. Adipose tissue is located under the skin and consists of closely packed adipocytes. Adipocytes are characterized by their rounded shape and cytoplasm filled with lipid droplets. Lipid molecules enter the adipocyte. Lipid molecules coalesce into a large droplet within the adipocyte. The accumulation of lipid causes the adipocyte to swell in size over time. Lipid release begins as the large lipid droplet within the adipocyte breaks down. The smaller lipid units are expelled from the cell, reducing the adipocyte's overall size. After lipid release, the adipocyte returns to its original size, completing the cyclical process.
Beta cells are distributed throughout the pancreas. This visualization emphasizes the spatial arrangement of beta cells. The model shows the collective behavior of beta cells within the pancreas. Beta cells exhibit rhythmic activity, illuminating in coordinated waves every few seconds. This rhythmic pattern indicates a synchronized insulin release mechanism. The synchronized insulin release is controlled by an internal biological clock. An internal clock graphic is synchronized with beta cell illumination. Timed meals influence the insulin secretion spikes, demonstrating the link between external stimuli and internal regulation. This synchronization illustrates the connection between external stimuli and internal biological processes.
Skeletal muscle fibers are visualized at the microscopic level, revealing the intracellular environment. GLUT4 transporters are distributed within the cytoplasm of muscle cells. These GLUT4 transporters are poised for mobilization upon stimulation. Elevated glucose levels or insulin signaling triggers GLUT4 transporter translocation from the cytoplasm. GLUT4 transporters are stored in vesicles that move towards the cell membrane. Vesicles fuse with the plasma membrane, increasing GLUT4 concentration and facilitating glucose entry. The muscle's peripheral clock influences glucose uptake efficiency. Glucose uptake varies according to circadian rhythms. Circadian rhythms modulate insulin sensitivity and metabolic regulation within the muscle.
The hypothalamic clock pulses rhythmically, indicating its role in regulating bodily functions. The hypothalamic clock plays a key role in the timing of feeding cues. This internal clock synchronizes with external stimuli, influencing appetite and metabolic processes. The vagus nerve transmits signals from the brain to the gut. These signals coordinate digestive processes, including gastric motility. The vagus nerve also regulates enzyme secretion and nutrient absorption. The vagus nerve sends signals to the gut, initiating physiological responses. These signals trigger the release of hormones and the activation of digestive enzymes within the gut. This bidirectional communication ensures efficient nutrient processing and energy homeostasis.
The adrenal glands are positioned superior to the kidneys. Each adrenal gland emits a soft, golden glow, indicating its active state. The pulsing glow represents the cyclical nature of cortisol production. Cortisol release is visually linked to the circadian rhythm. The timing of cortisol secretion is emphasized by the 24-hour cycle. The clock face visually integrates with the adrenal glands. Metabolic processes are visually interconnected with the adrenal glands and a circadian clock. Activation of the adrenal glands and progression of the circadian clock correlate with changes in metabolic activity. Cortisol, released by the adrenal glands, exerts a significant influence on metabolic functions.
Brown adipose tissue contains a high concentration of mitochondria within its cells. Mitochondria are essential for ATP production and thermogenesis in brown fat. Cristae, within mitochondria, play a crucial role in energy production. The electron transport chain pumps protons across the inner mitochondrial membrane, establishing an electrochemical gradient. This proton gradient drives ATP synthase to produce ATP, the cell's primary energy currency. In brown fat, UC-pone allows protons to flow back across the membrane without producing ATP, releasing energy as heat. Meal consumption triggers increased activity in brown fat mitochondria. During nutrient processing, fatty acids are released and activate UC-pone within the mitochondria. UC-pone activation leads to increased heat production, correlating with the timing of food intake.
The human body's internal organs gut, liver, pancreas, adipose tissue, and hypothalamus are visualized within a transparent form. Each organ features a clock face, visually representing its role in the body's circadian rhythm. All organ clocks tick in perfect synchrony, illustrating the body's natural circadian alignment. Organ clocks gradually drift out of sync as the simulation progresses. This desynchronization visually represents the disruption of the body's circadian rhythms. Irregular eating patterns can cause significant disruption to internal biological clocks. Timed eating facilitates the gradual realignment of organ clocks within the body. Consistent meal timing helps resynchronize the body's peripheral clocks. Resynchronization of peripheral clocks restores overall circadian harmony and metabolic efficiency.